In this lesson, we'll complete our discussion of the organelles within a cell by taking a look at the membranous organelles. If you remember from a previous lesson or our last lesson that we divide organelles into non-membranous organelles and membranous organelles. We had previously looked at the non-membranous organelles. So these that we're going to do in this lesson have their own membrane separating the organelle from the cytosol. We're going to take a look at the ER, known as the endoplasmic reticulum. Now there's two types. We're going to see there's rough and smooth. Then we'll take a look at the Golgi apparatus, lysosomes, peroxisomes, and mitochondria, not necessarily in that order. So let's take a look at the ER first. Now there's two types of ER. There's going to be rough, and if we go to the next slide for a minute, you'll see there's going to be smooth. The ER, first of all, before I define like what rough and smooth is, think of the ER as a membrane system. It's going to be a bunch of membranes that are folded and they coil through the cytoplasm. Right? So we have this membrane system enclosing this cavity known as the cisterna. I'll show you what I mean by that. So if you look over here, this is going to be a membrane, right? And see how it folds here, and then it folds over this way, right? And then it folds this way, forming this kind of hollow area in the middle. That is known as the cisterna. Now, this ER, this endoplasmic reticulum that we're looking at, has these little blue dots on it. If you remember from our previous lesson, these were the ribosomes. So since when we look at it, the ribosomes give it like a rough sandpaper appearance, we call this the rough ER. In our previous lesson, we had said that ribosomes could be attached to the ER. These were actually called fixed ribosomes. And what are they affixed to or attached to? Is the rough ER. So the rough ER is externally studded with ribosomes. Again, guys, that's what gives it the rough appearance. So now think about this from our previous lesson. If I was in class right now, I would say, hey, does anybody remember what's the function of the ribosomes, right? And you should automatically say in your mind, protein synthesis. So that means this endoplasmic reticulum, since it has ribosomes, has to be involved with protein synthesis, and that's exactly what it does. It's active in protein as well as glycoprotein synthesis. If you remember, glycoproteins is something we saw in previous lesson. It helps to fold the protein into secondary and tertiary structures. If you remember again from chemistry, secondary structures were the alpha helix, the beta sheet. The tertiary structure was how the whole protein folded on itself, and this is what was the active form of the protein. We're going to see that the ER then, the rough ER, is also then going to take these protein and put them into like a little truck known as the transport vesicle and deliver it to another organelle known as the Golgi apparatus. So think of the endoplasmic reticulum as being a factory. This rough ER, since it has ribosomes, what is this factory producing? Proteins. Then the factory is going to take those proteins, put them into a little truck known as a transport vesicle, and send them, right, ship them out to the next organelle that we'll look at in a minute, known as the Golgi apparatus. This I want you to think of as being like a warehouse, right? So the ER is a factory. The Golgi apparatus is going to be like a warehouse. We'll talk about that in a couple minutes. So just to give you an idea, this is a little bit advanced, but here's a ribosome. It's attached to the ER, right? So it's a rough ER. This messenger RNA, you can ignore this for today. We're going to learn this about this in a subsequent lesson. This is what's going to control protein synthesis. So if you notice here, guys, these little balls right here are amino acids bonded together, forming a protein. A polypeptide is a protein. And notice now how the protein is folding on itself, right? That's the tertiary structure, okay? So now we had the protein made, and watch this. The protein is going to go through the ER and go into this little balloon over here, like a water balloon, and we can see the protein in the water balloon. This will eventually pinch off and break away, forming that little truck known as the transport vesicle. And that's going to head up to the warehouse, which we'll talk about that in a little bit. Okay. All right, so the rough ER contains ribosomes. Anytime we have ribosomes, what's being produced? Proteins. Right? You can't go wrong with that answer. Now, there's also ER that does not have the ribosomes on it. So it's going to have a more smooth appearance, and this we're going to call the smooth ER or the SER. Since there are no ribosomes, guess what this factory cannot produce? No ribosomes, no proteins. So it is a factory though. What does it produce? Check this out. Some of the things we need for our cell membranes. Remember the phospholipids, right? The phospholipid bilayer, cholesterol that we looked at, right? So those are made for our cell membranes. In organs that produce steroids, like steroid hormones in the reproductive system, you may know testosterone, estrogen, the smooth ER would produce those hormones there. In our fat cells, it produces our glycerides, which are fats. And then in our liver and muscle, the smooth ER produces glycogen. But notice all of the things that are produced, none of it is proteins because there are no ribosomes. So I like to think of as the smooth ER producing everything else. Okay, so both endoplasmic reticulums are factories. Depending if there's ribosomes or not, that will determine if it's producing proteins. The rough ER has ribosomes, it produces proteins. The smooth ER has no ribosomes, so it can't produce proteins, but it produces all these other important things: phospholipids, cholesterol, steroid hormones, and again in glands that produce these hormones, fats, glycogen. Right, so here's a picture of the rough ER. Notice it's close to the nucleus. We'll talk about that in a subsequent lesson. So all of these little dots right here are the ribosomes. But notice out here, guys, the ER has no ribosomes, has a more smooth appearance. Guess what we're going to call that? The smooth ER. Right? And then in both cases, whether it's the smooth ER, the hollow area in the middle, right here is a good one, all this hollow area in the middle, that's the cisterna. That's just the place where the proteins would flow, or the phospholipids or the cholesterol over here would flow. So let's pretend for a minute we had the rough ER produce a protein, right? It's a factory. It sends the protein into the little truck called the transport vesicle. That little truck drives up the block to the warehouse. The name of the warehouse, and I'm going to show you a picture in a minute, is known as the Golgi Apparatus. Right? Some people call it the Golgi complex. This is also membranes that are forming these little, sacs and these little hollow areas in the middle. Okay, so it's very similar to the ER in structure. Let's take a look. So if you notice, this is where the endoplasmic reticulum was, next to the nucleus. Right? A little bit further away, these folded membranes right here, all of these folded membranes are going to be the Golgi apparatus. Right? They're going to be the Golgi apparatus. Now, those transport vesicles that came from the endoplasmic reticulum are going to enter the Golgi apparatus at something known as the forming phase. So what do I mean by that? So watch this, guys. These little bubbles over here, these are the little trucks that were coming from the endoplasmic reticulum. They drive up and they fuse with this membrane over here. This would be called the forming phase. And now whatever's inside this little water balloon or this little truck known as the transport vesicle is going to go into the Golgi apparatus, and it's going to go through this Golgi apparatus. And we're going to see that the Golgi apparatus may modify that protein, repackage it, and then the Golgi apparatus will send it out itself. So this face right over here is known as the forming phase. Right? We're going to call that the forming phase, and that's where the trucks again, the transport vesicles are dumping the material into the Golgi apparatus over here. Okay, so let's go back for a minute. All right, so these, if you want to just make this clear, the transport vesicles, they enter the forming phase. And then we're going to see that whatever leaves the Golgi apparatus is going to leave on the other side, known as the maturing phase. So this side over here is going to be the maturing phase, and I'll show you that in a minute. So the Golgi apparatus does a couple important things for us. Number one, it's gonna, whatever it receives from the endoplasmic reticulum, it may modify and repackage these secretions. So things like hormones, enzymes, right? They're gonna be repackaged and modified in the Golgi apparatus. It may also modify the proteins that are coming in. It may add a carbohydrate. It may take carbohydrates away, but it's gonna kind of remodify or modify the proteins. And then we're also going to see, and I'll show you this in a minute, it produces lysosomes and it helps to fix plasma membranes that have been damaged. Right, so let's take a look what I'm talking about. Here's an actual picture of the Golgi apparatus here. They have to magnify this 42,000 times. Okay, so put your pens down for a second. So watch this. This on the left is the nucleus. The nucleus controls protein production. This is a little bit advanced. Don't worry about that right now. But it sends out this green line right here. This is the messenger RNA that's going to build the protein at the ribosome. So notice, guys, here's the ribosome. This is where I want to start this story. The ribosome produces these amino acids here. So this is the beginning of our protein. And see how the protein gets folded like this? All right, so we just made our protein. It goes through the rough ER, and then it's going to go into this thing here, like a little water balloon that's going to pinch off. This is going to be the transport vesicle. Okay, so that's like the little truck that's going to take the protein and drive it up the block. So here it drives up the block to the warehouse. Notice this transport vesicle, see what it does? See how it fuses with the Golgi apparatus. This is known as the forming face over here. Right, this is the forming face. This is what's receiving the transport vesicle. Now the protein is inside the warehouse, and the protein, guys, look at this, goes through the warehouse. But notice the colors of the protein are changing, right? Originally here it was all kind of pink little amino acids, but now I see blue and pink. So that means things are changed. That tells me that the Golgi apparatus, our Golgi apparatus modified the protein. So watch this. Let's just say now here is the modified protein. It's going to also go into a little truck. It's like a water balloon, but instead of having water in it, we're going to have the protein. This is going to pinch off. This truck is called the secretory vesicle. Remember the one before was called the transport vesicle. That's the one that went to the Golgi apparatus. The one that's going to leave is known as the secretory vesicle. And this face here, like the exit, is called the maturing phase. Right, so we have the forming phase over here and the maturing phase over here. Now, this protein, in this case, we want it to leave the cell. Watch what it does. This cell secretory vesicle is going to travel to the cell membrane, fuse with the cell membrane, and check this out. It's going to almost like spit out the protein into the fluid around the cell, and now this protein can go into the blood and go somewhere. Let's just say this is an example of the pancreas right here, a cell in your pancreas. You guys have heard of insulin. This is the protein insulin, let's say, that was made here. Then we modified the insulin to its final product. Now the pancreas cell is going to get rid of it, put it into the bloodstream so that it can lower your blood sugars, let's say. Okay, this process of releasing something from the cell, this is important. This is known as exocytosis. Exo means to exit. Cyto means the cell. And I love this picture here. Here's an actual picture. Here's the secretory vesicle. And then look at this. Here's the protein being spit out into the extracellular fluid. Okay, so what happened is the protein was made in the factory known as the rough ER. The protein got shipped to the warehouse into the forming phase by the transport vesicles. The protein goes through the warehouse, it gets modified, and we could tell, how do I know? It looks different than when it came in, right? So it went through, now it's the final form of the protein. It goes into this little water balloon known as the secretory vesicle. That's at the maturing phase. Then the secretory vesicle drives to the cell membrane, and that secretory vesicle fuses with the cell membrane and spits out that protein. That process, I want you to know, is called the exocytosis. Before I go back to the notes, watch this. Let's say another protein comes in, it goes through, but this one is repackaged into a different protein. How do I know it's different? It's kind of like orange amino acids and green right here. This will pinch off, but it's not going to be called the secretory vesicle. This is actually going to pinch off and give rise to one of the organelles that we're going to talk about in a minute, known as the lysosome. So the Golgi apparatus creates our lysosomes. Okay, the third thing, let's just say a cell membrane was damaged, like there was a hole poked into the cell membrane here. We can repair that, kind of like if you had a hole poked into your wall, right? You could spackle it, sheetrock it, spackle it, sand it, and paint it, and never know there was a hole there. Well, watch what happens. Let's say there was a little hole punched into the cell membrane here. The Golgi apparatus can take the membrane of the Golgi apparatus, but watch this, with nothing in it, ship this membrane vesicle known as a membrane renewal vesicle, ship this over and patch up the hole, because again this vesicle here is made up of cell membrane, right? It's a phospholipid bilayer, and it can patch up this hole right here. So we could see the Golgi apparatus from this picture has three important functions, right? It'll ship out a protein via exocytosis, it produces our lysosomes, and it can also repair cell membranes. Okay. If you want to even add here, release from the cell, just put in parentheses. I didn't put it here, by exocytosis, right? Release from the cell by exocytosis. All right. It can renew, excuse me, or modify the plasma membrane. That was this right here. This fixing this hole here, or it can make the lysosomes, which we're going to talk about in a minute, and it produces the lysosome over here. Okay. Here's just another picture, kind of giving the same idea. Again, here's the endoplasmic reticulum. Here's your transport vesicles driving up to the forming phase. Then the protein goes through the Golgi apparatus and comes out the mature phase. Then these are the secretory vesicles that are going to bring the protein and by exocytosis spit out the protein, right? So that's one way of getting rid of a protein. Here it's showing you the Golgi apparatus repairing a cell membrane, right? There's a little hole in the cell membrane right here. And then here it's showing you how it's producing the lysosomes. Okay, so it's just another picture giving us the same information. Okay, last couple organelles. The next organelle is also important. We're going to look at this in more detail later in the semester. This is known as the mitochondria. You could think of this as the powerhouse of the cell. This is where we produce our energy known as ATP. Right? We looked at this in chemistry, adenosine triphosphate. So this is a rod-like membranous structure. It has a double membrane. If you kind of look at it right here, this is the mitochondria, and you'll see it has a double membrane. There's an outer membrane and then an inner membrane. You can see the double membrane here, right? So the outer membrane, and then here's the inner membrane right over here. So it's got two membranes. The outer membrane is smooth, and then the inner membrane has these folds. If you take a look, all these folds right here, these folds are known as the cristae. And then this would be filled with like a fluid in here. That's known as the matrix. We have it labeled over here, right? So that fluid in here would be the matrix. All right, so the outer membrane is smooth. The inner membrane has numerous folds. We call those folds cristae. And then the fluid that's, kind of inside the cristae is known as the matrix. This is where all the chemical reactions take place to produce ATP. So that's the main thing I want you to know about the mitochondria. It is known as the powerhouse of the cell. If I were to ask you on an exam, what does the mitochondria do? Right here, produces energy, right? And we're going to see we can actually burn glucose to make ATP. And I'll show you how that's done down later in the semester. So here's a real picture. Look at this, 50,000 times this was magnified to show you the mitochondria. And this is just a little bit advanced, but just to kind of give you an idea, guys, if we had glucose, it would go through a series of 10 chemical reactions over here to become 2-pyruvate molecules, right? So we're splitting glucose into 2-pyruvate. That goes into the mitochondria, and then it goes through a series of chemical reactions known as the citric acid cycle. Right? You may have learned it as the Krebs cycle. You may have heard it as that. And then it'll go through something known as the electron transport chain and oxidative phosphorylation. Again, this is a little bit advanced, but look what comes out. This is the main thing. What are we producing? ATP. Okay, and you'll notice the mitochondria only work if we have oxygen going into the mitochondria. As long as we have plenty of oxygen, the mitochondria will produce a lot of ATP. And I'll talk about that again later in the semester. I'm not too concerned about these chemicals right now or the citric acid cycle. Just know that the mitochondria at this level produce ATP. Okay. The last two organelles that I want to show you are kind of like the digestive system of the cell in my mind, right? They're kind of break things down. We're going to look at lysosomes and peroxisomes. So the first dot we know already. These are powerful containing vesicles produced by the Golgi apparatus, right? I showed you that a little while ago. So that's where they're made. Now, when a lysosome is made and it's not doing anything, it's just kind of hanging out in the cell, we call it a primary lysosome. Right? It contains inactive enzymes. Right? So it's known as a primary lysosome. But if we activate the lysosome to break something down, then we're going to change the name to a secondary lysosome, right? So these are formed when the primary lysosome fuses with a damaged organelle, as an example, and the enzymes are activated. So let's just say, as an example, a mitochondria inside the cell becomes worn out. Right? It's not functioning properly. What will happen, guys, and I'll show you a picture in a minute, is the lysosome will attach to the mitochondria. Now it becomes activated. We call it a secondary lysosome. Okay, so these lysosomes will destroy bacteria. They'll break down molecules. Again, think of it like the digestive system. And they can even break down worn out or damaged organelles. Now, we actually have a term for that called autophagy, right? AUTOPHAGY, right? That's a term we use when we recycle organelles or break down organelles that we don't need. Okay. One other thing before I show you a picture. Sometimes we have cells, maybe too many cells in the body, or a cell becomes inactive or it's damaged, and we want to get rid of the entire cell. So this is interesting to me. It's like the cell commits suicide. We call this autolysis. Auto means self, and lysis means breakdown. Right? So it actually, the cell is killing itself. It's committing suicide. What happens is all the lysosomes in the cell release their digestive enzymes into the cell, killing the cell. All right, so it's a way of the body getting rid of cells that it does not need. We call that autolysis. All right, so here's just a picture showing you the lysosomes. In this picture, they happen to be green. So let me just show you real quick, just to kind of give you an idea of how the lysosome works. So hopefully you recognize, here's the Golgi apparatus. And remember we said that the Golgi apparatus gives rise to the lysosome. So when the lysosome is just hanging out in the cell, not activated, what are you going to call it? Primary lysosome. But let's just say we have this mitochondria over here that's worn out or damaged. We don't want things that are worn out or damaged. Watch what happens, guys. The lysosome will attach to the mitochondria, and now its enzymes become activated, and the enzymes go into the mitochondria, breaking it down. But what are you going to call this lysosome? secondary. All it means is it went from being inactive to active. Then the lysosome will continue to break down the mitochondria, and then check this out, we can actually get rid of the fragments of the mitochondria by, look at this process here, exocytosis. Okay, so that's an example of breaking down a worn out organelle. Another thing that we could do is this. Let's just say this cell, what we're looking at, is a white blood cell. White blood cells can bring in, let's say this was a bacteria as an example. Right, the white blood cell brings in the bacteria. Now the lysosome, which was inactive, attaches to this little vesicle with the bacteria in it, and it breaks down the bacteria, and we get rid of the fragments of the bacteria. So white blood cells have a lot of lysosomes in them also. But really right now I just want you to know primary is inactive, secondary is active. And again, the general functions, right? They'll break down bacteria, if you want to put as an example, white blood cells do that, break down molecules, break down damaged organelles. Like I said, we call that autophagy. If we have too many cells in the body, we could actually break down whole cells. That process is called autolysis, right? That's like the cell committing suicide. And again, here's just a good picture showing us a primary lysosome turning into a secondary lysosome in this case, or a primary lysosome turning into a secondary lysosome in this case. And in case number 3, they're actually showing you autolysis. Let's say we don't need this whole cell right here. The lysosome will break apart and release the enzymes into the cell and then kill the whole cell. Right? So they're showing you the different things that the mitochondria can do. Not mitochondria, lysosome can do. Sorry about that. Okay, the last organelle is kind of like the lysosome, except it won't do like autolysis. It won't do autophagy. Instead, the peroxisomes are going to break down more organic compounds, such as fatty acids. These are not produced by the Golgi apparatus. They're produced by other peroxisomes, right? So these, unlike the lysosomes, do not come from the Golgi apparatus. But these also contain enzymes, and again, they're going to break down more of like organic molecules such as fatty acids. Okay, they'll even do detoxification, right? If you detoxify, they can help to break down toxic substances. As an example, hydrogen peroxide, if that were to build up in the cell, that can be damaging. So this will help to break that down. Okay, so that concludes this lesson. Here's just a picture of the peroxisomes over here. That concludes this lesson on the membranous organelles, right? So now we have them all down. We have the non-membranous organelles as well as the membranous organelles.